Welcome to Deep Dive into Knowledge.
Why do we grow old? The science behind aging.
Welcome to a deep dive into knowledge.
Thank you.
I want to ask you, the listener, to do something for me, just for a moment, wherever you are. I want you to sit completely still. If you are holding your phone, let your hand rest.
Right.
Or if you are driving, just settle into the seat. Take a breath, hold it for a second, and then let it go.
Yes.
In this moment of stillness, you likely feel a sense of continuity. You feel static. You feel like a solid object occupying space. You feel like the same person you were 5 minutes ago or 5 years ago. There is this sense of permanence to the experience of being you. You feel like a statue that breathes. But this sensation, this feeling of stillness is a lie. It is perhaps the most convincing cognitive illusion our brains have ever constructed, designed specifically to keep us sane.
Yes.
Because if you could actually feel what was happening inside your body right now, in this very second, it wouldn't be peaceful. It would be terrifying.
It would.
While you listen to the sound of my voice, a violent dismantling is occurring in the dark. It is happening in the deep layers of your skin, in the filtration systems of your kidneys, in the lining of your gut. Right now, thousands of your own cells are hurtling toward a terminal line. Some of them are dying at this exact moment, silently dismantling their own machinery and vanishing.
Exactly.
But others are doing something far more unsettling. They are turning into something, well, something different. They are starping. They are refusing to die, but they are also refusing to work. They are becoming stagnant, toxic entities that sit inside your tissues and rot from the inside out.
Yes.
We have a word for this. We call it aging. But that word feels, I don't know, too soft. It suggests a gentle fading, a cosmetic change, maybe a few gray hairs. What is actually happening is a mathematical subtraction.
Right.
It is a relentless mechanical countdown that was written into your genetic code the moment you were conceived. And it poses a question that we rarely stop to ask simply because we are too busy living through it.
Yeah.
Why does the machine break? If nature can build a human being from a single microscopic cell, an engineering feat of infinite complexity, why can it not simply maintain that machine?
Quite.
Why is there a mandatory expiration date? Why is the system designed to fail?
It is a profound paradox.
To understand this, we have to look at the invisible timer ticking inside every single one of us. So I want to start with the concept of the limit. For a long time, we didn't think there was one, did we? We thought life was essentially magic.
We did, yes. If you look at the scientific consensus in the early 20th century, it was actually quite optimistic. And looking back, perhaps a bit arrogant.
How so?
Well, the prevailing belief was that life at the cellular level was inherently immortal.
Immortal?
Yes. The idea was that the cells themselves didn't have an expiration date. It was only the container of the body that wore out due to external factors like disease or trauma.
So the assumption was that if you took a cell out of the body, away from the stress of the world, gave it perfect food and perfect warmth, it would just keep dividing forever.
Precisely. And this belief wasn't just a hunch. It was driven by one of the most famous and, as it turns out, flawed experiments in the history of biology.
Okay.
In 1912, a Nobel Prize-winning surgeon named Alexis Carrel began an experiment involving a culture of chicken heart cells.
A chicken heart?
Yes. He placed these cells in a nutrient broth and claimed to have kept them alive and beating for decades.
Decades? I mean, that is longer than a chicken actually lives.
Much longer. Carrel kept this culture going for over 30 years. It became an absolute sensation.
Wow.
It fueled the idea that the cell is immortal. It suggested that death was not a biological necessity, but just a failure of the environment. People genuinely believed that if we could just perfect the environment, we could live forever.
But that wasn't true. The bricks weren't magic. The house was just being propped up. What actually went wrong with that experiment?
It was a simple, very human error. Carrel and his assistants were feeding the cells a sort of chicken embryo extract every day to provide nutrients. What they didn't realize, or perhaps what they refused to admit, was that the extract itself contained fresh, new cells.
Oh, I see.
They were unknowingly adding young cells to the dish every single time they fed it.
Right.
They weren't sustaining an immortal culture. They were constantly replenishing it.
So the immortality was a mirage all along, and it took nearly 50 years for someone to actually prove it.
It took until 1961. A young microbiologist named Leonard Hayflick finally shattered the illusion.
What did he do?
Well, he wasn't even trying to study aging initially. He was just trying to grow normal human fetal cells for other research. But he noticed something that Carroll had missed or ignored. Hayflick observed that no matter how perfectly he treated the cells, no matter how much space or food they had, they would divide for a while, and then they would slow down.
They would look ragged.
Yes, they looked ragged under the microscope. And then they would just stop.
They hit a wall.
They hit a hard mathematical wall. Hayflick found that normal human cells will divide approximately 50 times.
Just 50.
Just 50. That is the limit. It became known as the Hayflick Limit, roughly 50 population doublings.
And then what happens?
Once a cell line hits that number, the game is over. They enter a state of permanent arrest. They can never divide again.
50 times. When you say it like that, it sounds incredibly small. We lived for 80 years. How does 50 divisions sustain an entire human life?
It is the power of exponential growth. If you take the number 2 and double it 50 times, you get a number in the quadrillions.
Right, the math scales up quickly.
Exactly. It is enough to produce a full human body and maintain it for a significant period. But it is finite. It is a fuel tank that does not refill.
So Hayflick proved the limit existed, but he didn't know why. It took us decades to find the actual mechanism, the physical counter inside the cell. And this brings us to telomeres. I've always heard the shoelace analogy, but I feel like we need to go deeper than that. What is actually happening at the molecular level?
The shoelace analogy is the best starting point, certainly, but the biochemistry is where it gets fascinating. Think of your DNA, your chromosomes, as long strands containing all your genetic data. At the very tips of those strands are repeating sequences of non-coding DNA.
These are the telomeres.
Yes. Now here is the design flaw. The enzyme that copies our DNA polymerase, cannot copy the very, very tip of the strand.
Why not?
It is called the end replication problem. Imagine painting a floor, but you paint yourself into a corner. You physically cannot paint the spot you are standing on.
I see. So every time a cell divides, the copy comes out slightly shorter than the original.
Exactly. The molecular machine cannot grab the very end. So with every division, a small chunk of that telomere buffer is shaved off.
Like a fuse burning down.
Very much like a fuse. And when the telomere gets too short, the protective cap falls off and the raw DNA is exposed.
Which is bad.
It is catastrophic for the cell. The cell detects this as a massive genetic break, like a snapped chromosome, and it pulls the emergency brake. It stops dividing entirely to prevent genetic chaos.
This implies a terrifying transaction. It means that every time my body does something good, every time it heals a cut or replenishes my blood or grows new skin, it is paying a permanent price.
That's the trade-off. It is a biological currency. If you slice your finger while cooking, your skin cells must divide to close that wound.
And that costs something.
It costs you telomere length. Your intestinal lining replaces itself every few days because digestion is a harsh process. That turnover costs telomere length. You are literally burning through your fuse just to stay intact.
It really reframes the concept of living. We aren't just existing, we are spending. And this isn't just theory anymore, is it? We can actually see this correlation in the population.
We can. There was a significant study published in 2024 that linked telomere length directly to lifespan predictions.
What did the data show?
The data is stark. If you look at mice, or even humans, those with shorter telomeres for their age bracket tend to have shorter remaining lifespans.
They are biologically older?
Yes. They are more susceptible to disease. It is a biological countdown clock that starts ticking the moment you are conceived.
That feels incredibly fatalistic. It sounds like we are just batteries running down, and once the charge is gone, we stop. But the research suggests it is not just the passive ticking of the clock. We are also actively participating in our own destruction.
That is true.
We aren't just running out of time, we are breaking the machine. And the irony is that the primary culprit is the very thing we need to survive: oxygen.
This is the oxygen trap. It is one of the great ironies of biology. We are aerobic organisms.
Meaning we need oxygen to live.
We need oxygen to burn fuel glucose to create energy. That process happens in our mitochondria, the power plants of our cells. But the process of burning fuel with oxygen is dirty.
Dirty in what sense?
In the chemical sense. The electron transport chain, the assembly line inside the mitochondria that makes energy, is not 100% efficient. It leaks.
Leaks what?
Electrons. Occasionally, electrons escape the assembly line and latch on to passing oxygen molecules, creating what we call reactive oxygen species, or ROS. You might know them better as free radicals.
I think most of us have heard the term free radicals in marketing for skin creams or health drinks. But what are they actually doing inside the cell?
They are molecular vandals. A free radical is an atom or molecule that has an unpaired electron. It is chemically unstable.
So wants to find a partner.
It is desperate to find a partner for that electron. So it will rip one away from anything nearby. It will violently tear electrons off your DNA, your proteins, your cell membranes.
So it is effectively rusting.
It is chemically identical to rusting.
Yeah.
Oxidation is what turns iron into rust. We are slowly rusting from the inside out.
But surely the body has a defense against this.
It does. We evolved with free radicals, so we have built-in defense systems. We produce antioxidant enzymes that are constantly sweeping up these free radicals.
But they can't catch them all.
No, the system isn't perfect. And as we age, the cleanup crew gets slower and the damage accumulates.
This leads to a common misconception, doesn't it? If free radicals are bad, then antioxidants must be good. So logically, I should just take massive doses of vitamin C and vitamin E and I will live forever. The data doesn't support that.
No, it doesn't. And in fact, high-dose antioxidant supplementation can sometimes shorten lifespan.
Why is that?
This is where the nuance is critical. We evolved with free radicals. Biology is highly efficient. It uses everything. It turns out the body actually uses these reactive oxygen species as signaling molecules.
They are messengers.
They are the smoke alarm. When a cell is under stress, a burst of ROS tells the nucleus, hey, we have a problem here, send help.
Oh, I see.
If you completely wipe out all free radicals with massive doses of artificial antioxidants, you silence the smoke alarm. You blind the cell.
It doesn't know it is under attack.
Exactly. So it doesn't repair itself. It is not a simple battle of good versus evil.
Yeah.
It is a delicate balance.
Yeah. So we have the hard limit of the telomeres getting shorter, the fuse burning down, we have the damage from the oxygen, we breathe the rusting of the machine. But then there's this third concept, and I have to say, this is the one I found the most disturbing.
Yes, it is quite profound.
We mentioned earlier that when a cell hits its Hayflick limit, it stops dividing, but you said it doesn't always die. It becomes something else. It enters a twilight state.
Yes. This is the concept of cellular senescence.
Mm.
And in the scientific field, we have a very descriptive nickname for these entities. We call them zombie cells.
A zombie cell. That sounds like science fiction, but you mean it quite literally.
It is the most accurate biological description we have. These cells are not dead. They are metabolically active.
They're still eater.
They are eating nutrients, yes. They're taking up physical space,
Mm-hmm.
but they are not functioning. They have shut down their primary job. They are stuck in a state of animated suspension.
But if they just sat there and did nothing, that wouldn't be so bad. It would just be dead weight. But they aren't passive, are they? They are dangerous.
They are incredibly dangerous. A senescent cell is not a benign retiree. It changes its personality completely. It begins to secrete a toxic cocktail of chemicals.
What kind of chemicals?
We call this the SASP, the senescence-associated secretory phenotype.
That is quite a mouthful. What is in this cocktail?
It is a harsh mix of inflammatory cytokines, proteases, and growth factors. Things like interleukin-6. These are distress signals that trigger inflammation and tissue degradation.
So they are constantly sounding the alarm.
Imagine a person in a quiet cinema who suddenly starts screaming, fire? Constantly, without stopping. That is a senescent cell.
And what does that screaming do to the healthy cells sitting next to it?
This is the horror of it. It corrupts them. It creates what we call a bystander effect.
Contagion.
Yes. The inflammatory signal from the zombie cell can actually drive neighboring, perfectly healthy cells into senescence. It's contagious. One zombie cell infects its neighbors, creating a growing cluster of dysfunction. It spreads a rot through the tissue.
So as we age, it is not just that we have fewer working cells. It is that we are slowly filling up with these toxic, screaming zombies that are poisoning the well.
Exactly. When you are young, your immune system is crisp. It spots a senescent cell and eliminates it immediately.
But the immune system ages too.
It does. As you age, your immune system weakens partly due to telomere shortening in the immune cells themselves. So the zombies start to accumulate. They build up in your joints, your arteries, your brain.
And this accumulation, this rising tide of toxicity is what drives inflammaging.
Inflammaging, yes.
Yeah.
Chronic, low-grade sterile inflammation is the engine behind almost every age-related disease we face. Atherosclerosis, Alzheimer's, arthritis.
So these diseases aren't just random wear and tear.
No, they aren't just things that happen. They are the direct result of this cellular screaming.
I want to ground this for the listener. We have been talking about microscopic mechanisms, telomeres, ROS, SASP, but how does this translate to the human experience? When I look in the mirror and see a wrinkle, or when I feel my knees ache, or when I can't read the menu in a dark restaurant, connect the dots for me. How does cellular failure become systemic failure?
It is a scaling problem. Let us look at wrinkles, the most visible sign of aging. Your skin relies on cells called fibroblasts to produce collagen and elastin.
The structural proteins.
Yes, the proteins that keep skin firm and snappy. As those fibroblasts hit their Hayflick limit or become senescent due to UV damage from the sun, they stop producing collagen.
They just shut down.
Worse than that, the senescent ones release enzymes that actually eat the existing collagen. The scaffolding collapses. The skin sags and folds.
What about energy? The fatigue that older people feel, that sense that the battery just doesn't hold a charge anymore?
That goes back to the mitochondria. As they accumulate oxidative damage from the free radicals over the years, they become inefficient. The voltage drops.
So the engine loses power.
If your muscle cells have damaged mitochondria, they cannot produce ATP, the energy currency, fast enough. You try to run up the stairs, but the engines simply cannot generate the horsepower. It is not a lack of willpower. It is a lack of molecular fuel.
And digestion. That seems to be a major complaint as we age.
The lining of your gut is one of the most dynamic tissues in the human body. It completely replaces itself every few days.
Because it's a harsh environment down there.
Very harsh. That rapid turnover requires a massive amount of cell division from local stem cells. As those stem cells exhaust their telomeres over the decades, the repair slows down. The barrier becomes weaker.
Which means things get through that shouldn't.
Exactly. Nutrients aren't absorbed as well. You develop leaky gut, which lets more toxins into the bloodstream, causing more systemic inflammation, which then creates more senescent cells. It is a vicious, accelerating cycle.
A symphony of dysfunction. Every instrument playing slightly out of tune.
And eventually, the noise drowns out the music entirely.
Up to this point, this conversation has been quite bleak. We have described a machine designed to break, a fuse designed to burn out, and a toxicity that accumulates inevitably. If we stopped here, the takeaway would just be, accept your fate.
Yes, it would.
But that is not where the science is right now. We are living through a pivot point in history, a moment where we stopped just observing the decay and started asking if we could treat it.
It is an incredibly exciting time.
I want to talk about the mouse study,
Yeah.
the one from 2016 at the Mayo Clinic. This feels like the moment everything changed.
The Baker study, yes, this was the absolute earthquake in the field. Researchers asked a very simple, elegant question.
What was it?
They asked, if senescent cells, these zombies, are driving aging, what happens if we just kill them?
Just clear them out.
Right. They genetically engineered a strain of mice so that their senescent cells could be eliminated on command using a specific drug trigger. They let the mice get old naturally.
And then?
One group was left alone as the control. The other group had their zombie cells flushed out.
And the visual difference was shocking. I have seen the photos.
It is undeniable when you look at them side by side. The control mouse looks old. It has a hunchback, gray thinning fur, cataracts, and it is moving very slowly.
And the treated mouse?
The treated mouse, the one without the zombie cells, looks vibrant. It has a thick, glossy coat, clear eyes, high energy, and it is actively running on its wheel.
They didn't just live longer, they stayed young longer.
That is the crucial key. They extended the health span, they delayed the onset of cancer, they delayed heart disease, they delayed kidney failure.
So it wasn't just cosmetic.
No, it was fundamental. It proved that senescent cells are not just a passive side effect of aging, they are a primary driver of aging. And if they're a driver, they are a target.
This launched the entire field of senolytics, drugs that kill zombies. And we aren't just talking about genetic engineering in mice anymore. We are talking about actual pharmaceuticals.
We are. The most famous combination currently being studied is D plus Q, dasatinib, and quercetin.
It sounds like a strange cocktail. One is a heavy-duty drug. The other is something I can buy at a health food store.
It is an odd couple, certainly. Dasatinib is a powerful chemotherapy drug used for leukemia. It is a hammer. Quercetin is a plant flavonoid found in apples, onions, and capers. It is much milder.
So why combine them?
Researchers found that when you use them together, they have a profound synergistic effect. They hunt down senescent cells and disable the specific survival pathways that keep them alive.
They pull the plug on the zombies.
Yes. They force the zombie cells to commit cellular suicide, a process called apoptosis, while leaving the healthy functioning cells entirely alone.
And the administration of this is interesting. It is not a daily pill like a vitamin. It is more of a hit and run.
Exactly. You do not want to be taking chemotherapy drugs every day.
Right.
But with Senolytics, you do not have to be. You just need to go in, wipe out the accumulated population of senescent cells, and then get out.
Clear the brush.
The tissue is clean. It takes weeks or even months for the zombies to slowly build up again.
Has this been tried in humans, or are we still just curing mice?
We are actively moving into human trials. There was a pilot study recently involving patients with idiopathic pulmonary fibrosis, a severe scarring of the lungs linked directly to senescence,
And what were the results?
and another trial with diabetic kidney disease. The results were preliminary, but very promising. We saw significant reductions in inflammatory markers in the blood. We saw actual improvements in physical function.
And there was a fascinating note about cognition in these studies. We tend to think of the brain as completely separate from the body, protected behind the blood-brain barrier. But clearing inflammation from the body seemed to help the mind.
The brain is not an island. Inflammatory cytokines, those screaming signals from the zombies in your belly fat or your liver, can cross the blood-brain barrier.
They get into the brain.
They do. They cause neuroinflammation, which manifests to the patient as brain fog or cognitive decline. By cleaning up the body's periphery, you lower the overall noise in the brain.
It sounds almost like an oil change. You flush out the sludge, and the engine runs smoothly again.
It is a very apt analogy, and researchers are looking for safer, more natural ways to do this long term.
Like what?
Inzatin, which is found in strawberries, is a potent senolytic. Theaflavins in black tea show promise. We are actively searching for molecules that can scrub our tissues gently over time.
So we have the potential to intervene. But how do we know if it is actually working? You can't just look in the mirror and ask, do I look less cellularly senescent today? We need a speedometer.
We need metrics, yes.
This brings us to one of the most important breakthroughs in modern biology, the clock. The idea that you actually have two ages.
This is the brilliant work of Steve Horvath and others. We now distinguish clearly between your chronological age and your biological age.
Chronological age being your birthdays.
Simply how many times the Earth has orbited the sun since you were born. That number is fixed. You cannot change it.
But the biological age is flexible.
The biological age is a measure of the actual wear and tear on your genome. And we can now measure it with incredible precision using what we call the epigenetic clock.
How do you read a clock that is written in blood?
You look at DNA methylation. To use a computer analogy, your DNA is the hardware, is the hard drive.
Right.
Epigenetics is the software, the operating system. Methyl groups are little chemical tags that attach to the physical DNA and tell the genes whether to be on or off.
So they control how the code is read.
Precisely. When you are an embryo, that pattern of tags is pristine. It is perfectly organized. But as you age, the pattern gets messy. It drifts.
Like corrupted software.
Yes. Some genes that should be turned off, like inflammatory genes, get accidentally turned on. Some genes that should be turned on, like repair genes, get turned off.
Okay, the software gets buggy.
It gets very buggy. And by reading these methylation patterns from a simple blood draw, we can tell you exactly how fast you're aging.
So the two ages can diverge.
Dramatically. You might be 50 years old according to your driver's license, but if you smoke, never sleep, and eat poorly, your methylation clock might say your body is biologically 60. You are sprinting toward the grave.
But if it is just software, can it be rebooted? Can we slow the clock down?
We can slow it down, and there is mounting evidence that we can even reverse it slightly.
Reverse it.
In clinical trials, using drugs like metformin, or even just aggressive lifestyle interventions, we have seen the methylation age drop. We can literally turn back the biological hands of time.
This is where I want to pivot to the practical. Because while we are all waiting for the senolytic pills to be FDA approved, the listener is aging right now. The clock is ticking while they listen to this conversation.
It is.
What can we do today? Not in 10 years, but today. What are the levers we can pull to slow down the countdown?
It is the part of the conversation where the science becomes incredibly boring, but also incredibly empowering.
The basics.
The magic pills currently available to us are lifestyle choices, but their effect on the molecular level is profound.
Let us start with exercise. We are always told to run to keep our hearts strong. But what is running doing to my telomeres?
This is the fascinating part. Exercise is not just about burning calories or building muscle. It creates a specific type of physiological stress that activates an enzyme called telomerase.
And what does that do?
This enzyme can actually add physical length back to the telomeres. High-intensity interval training, specifically, has been shown to boost telomerase activity. You are physically rebuilding the protective caps on your DNA shoelaces.
So exercise is literal genomic maintenance.
It is the most potent anti-aging drug we currently possess.
What about diet?
Diet is huge, but not just eating your vegetables. The most robust intervention, the one that consistently works in yeast, worms, flies, and mice, is calorie restriction.
Eating less overall.
Eating less, but strictly without malnutrition. When you constantly feed your body, it stays in growth mode. It focuses on dividing cells and building tissue.
Which burns the fuse faster.
Yes. But when you restrict calories, or when you fast, the nutrient sensors detect a shortage in the environment. The body switches gears into repair mode.
This is autophagy.
Autophagy, from the Greek for self-eating. When the cell is hungry, it stops dividing and starts looking around for internal snacks.
Internal snacks.
It finds broken proteins, damaged mitochondria, and bits of viral debris floating in the cell, and it recycles them for energy. It triggers a deep cleaning cycle. Fasting is effectively taking out the cellular trash.
Which brings us to the other time we take out the trash. Sleep.
Sleep is completely non-negotiable. For a very long time, we didn't fully understand why we needed to sleep so much. But recently, we discovered the glymphatic system.
Which is a plumbing system in the brain.
Literally. When you enter deep sleep, the brain cells actually shrink slightly in physical size, opening up tiny channels.
And fluid washes through.
Cerebrospinal fluid rushes in and washes through the brain tissue, physically flushing out the metabolic waste and toxins that built up during your waking hours.
Including the bad stuff.
Including the beta amyloid plaques, heavily associated with Alzheimer's disease.
So if I pull an all-nighter, I am leaving my brain marinating in its own sewage.
That is a graphic but highly accurate way to put it. Chronic sleep deprivation accelerates every single marker of aging. It prevents the repair crew from coming in for the night shift.
There's one more factor. We often separate the mind and the body, but you mentioned stress earlier. We know stress feels emotionally bad, but is it actually shortening my life?
It is. There was a landmark study on caregivers, specifically mothers looking after chronically ill children.
A very high-stress situation.
These women were under immense, unrelenting psychological stress. The study found that their telomeres were significantly shorter than healthy control groups. They had aged by an extra decade at the cellular level.
Their thoughts were influencing their DNA.
Exactly. Stress hormones like cortisol actively suppress the immune system and inhibit telomerase. And loneliness does the exact same thing.
Loneliness is a physical state.
Social isolation is as toxic to your cells as smoking 15 cigarettes a day. We are social primates. When we are alone, our bodies enter a threat state. Inflammation rises and repair downregulates. Connection is a biological imperative.
So if you want to slow the clock, move your body, stop eating all the time, sleep deeply, and call a friend.
It sounds simple, almost like folk wisdom.
Yeah.
But we now have the hard molecular proof.
But mere slowing isn't enough for some people. There are scientists who aren't satisfied with tapping the brakes. They want to put the car in reverse. They want to take the clock off the wall and wind it all the way back to zero.
Yes.
This sounds like the realm of fantasy, but we have to talk about the magic trick. We have to talk about Shinya Yamanaka.
This is the absolute frontier. This is the discovery that won the Nobel Prize and changed everything we thought we knew about fundamental biology.
What did he do?
In 2006, Shinya Yamanaka discovered that you could take an old adult skin cell, a cell that was fully committed to being skin, heavily burdened with all those epigenetic aging marks, and by exposing it to just 4 specific proteins, you could wipe its memory clean.
He turned an old cell into a baby cell.
He turned it back into an embryonic stem cell. He reset the epigenetic clock to absolute zero. The cell forgot it was old. It forgot it was a skin cell.
It became a blank slate.
It became pluripotent, capable of becoming any tissue in the body. These 4 proteins are now universally known as the Yamanaka factors.
So the obvious question is, why aren't we doing this to ourselves right now? Why can't I just take a Yamanaka bath and be 20 again?
Because there is a very, very fine line between youth and cancer.
Ah, that seems like a significant hurdle.
It is. An embryonic cell is designed to grow rapidly. It's designed to build a fresh body from scratch. If you indiscriminately reset all the cells in your body to an embryonic state, you wouldn't become a teenager again.
What happened?
You would become a giant mass of disorganized, rapidly dividing tissue. We call them teratomas.
The teratoma. That is a tumor, isn't it?
It is a monster tumor.
Mm-hmm.
Because the cells are pluripotent, the tumor tries to build random body parts wherever it is. You find teratomas containing fully formed teeth, hair, bone, even eye tissue. It is the stuff of nightmares.
That is horrifying. So we cannot just hit factory reset.
No. But the hope and the billions of dollars of investment currently flowing into this field lies in partial reprogramming.
Meaning you don't go all the way back.
The idea is to not go all the way back to zero. Just wind the clock back a little bit.
Make the cell younger, but make sure it still remembers it is a skin cell or a liver cell.
Exactly. You pulse the Yamanaka factors. You take a 60-year-old cell, scrub off the epigenetic noise so it functions like a 30-year-old cell, but you stop the process before it loses its fundamental identity.
Has this worked?
We have successfully done this in mice. Researchers took older blind mice with crushed optic nerves, an injury that simply never heals in adult mammals, and they applied partial reprogramming to the eye.
And the nerves heal.
The nerves regrew exactly like they were young embryonic tissue. Their vision returned.
That is staggering. It implies that the blueprint for a young body is still inside us even when we are old. We just lost access to the file.
The information is completely intact. We just need to learn how to safely restore the backup.
As we come to the end of this deep dive, I want to zoom out a bit. We have talked about the how, the telomeres snapping, the zombies screaming, the rust accumulating, but I want to ask the why.
It is the ultimate question.
If evolution is so good at selecting for survival, if survival of the fittest is the law of the land, why hasn't nature selected for immortality? Why are we programmed to die?
Because evolution does not care about you.
That is a blunt truth.
It is the hard reality of biology. Evolution only cares about the propagation of genes. It cares about the survival of the species, not the individual unit. There was a prominent theory called the disposable soma.
The disposable body.
Yes. Nature invests massive energy in building a strong, resilient body to get you safely to sexual maturity. It keeps you strong enough to reproduce and perhaps raise that offspring to independence.
And then?
But once you have successfully passed on your genes, you are, evolutionarily speaking, redundant.
We are discarded booster rockets, we get the payload to orbit, and then we fall into the ocean.
Precisely. Investing precious metabolic energy to maintain an old body forever is a waste of resources. From an evolutionary perspective, it is actually better for the old generation to break down and die, to stop competing for limited food, and to make room for the new generation with fresh genetic combinations.
So we are programmed to become obsolete. It feels almost like a law of physics, like entropy.
But here is the magnificent catch. It isn't a law of physics. It is merely a biological choice.
There are exceptions.
There are animals that do not play by these rules. Take the bowhead whale, it lives over 200 years, or the Greenland shark, which can live for 400 years without any signs of cellular decline.
What about the jellyfish?
The most famous example, yes. The immortal jellyfish, Turritopsis dohrnii.
The jellyfish that Benjamin buttons itself.
Exactly. When it gets old or injured or stressed, it literally reverts back to its juvenile polyp stage. It transforms from an adult back into a baby and then grows up all over again. It can do this endlessly. It is biologically immortal.
So aging is not inevitable. It is not like gravity. It is just code.
It is just code. And if it is code, it can be hacked. It can be rewritten. We just need to learn the language.
That is a profound place to land. It changes the way I look at the future entirely.
I think if we zoom out, there is a deep philosophical weight to all of this. We spend our lives terrified of this process. The gray hair, the slowing down, the loss of capability. It feels like a tragic failure.
It does.
And the suffering, the disease, the dementia, the physical pain that is a tragedy to be solved. We have a moral obligation to solve it. But the finiteness of life itself, the fact that there's a strict deadline, that might be the very thing that generates meaning.
Oh, so?
If we lived for 1,000 years, would we feel the burning urgency to write that book? To say, I love you, to forgive someone.
The scarcity of time is what gives it value.
Perhaps. So the ultimate goal shouldn't be to live forever in a state of stagnant preservation. Perhaps the goal is to fundamentally shift our focus from lifespan, simply counting the years on a calendar, to health span.
Quality over quantity.
The goal is to die young, as late as possible, to live a vibrant, active, perfectly healthy life right up until the very end, and then simply turn off the lights. That is the true promise of this science. Not immortality, but vitality.
We have traveled a long way in this hour. From the hard limit of the Hayflick number to the messy reality of the zombie cells inside us. We have moved from the inevitable decay of the machine to the newfound hope that this is a treatable condition, not a curse. I invite you, as you move through the rest of your day, to feel your body differently. Do not feel it as a decaying statue. Feel it as a dynamic process. It is a battleground, yes.
Okay.
It is dismantling and rebuilding itself in every single second. But it is a process you can influence.
Absolutely.
The clock is ticking, loudly and relentlessly, but for the first time in human history, we are learning how to read the time. And perhaps, soon, we will learn how to wind it back. Join us next time. We will be exploring another fundamental layer of our reality, deep dive into knowledge.